Welcome to our channel Mootechie. In this video, we will learn about factors affecting growth of bacteria. Microbiology overview. The main factors we'll explore are temperature, pH, oxygen, water activity or osmotic pressure, and nutrient availability. You can see them summarized here in this table: temperature, pH, oxygen, water activity, and nutrients. Each of these environmental conditions plays a critical role in determining whether bacteria can grow, how quickly they multiply, and where they can survive. Effect of temperature. Bacterial growth has minimum, optimum, and maximum temperatures. The growth rate peaks at the optimum. This means that for every bacterial species, there's a temperature sweet spot where it grows fastest, and going too far below or above that range will slow or stop growth entirely. Ranges are categorized. Psychrophiles grow between 0 and 15 degrees Celsius, mesophiles between 20 and 45, and thermophiles between 50 and 80. These groups have adapted their cellular machinery to function best within these specific thermal windows. Temperature shifts alter enzyme kinetics and membrane fluidity. Enzymes work at certain speeds depending on temperature, and the cell membrane needs to remain fluid enough for transport. Too cold it solidifies, too hot it becomes leaky. Cells induce heat shock and cold shock proteins to stabilize functions. These are like emergency repair crews that help protect proteins and maintain cell integrity during sudden temperature changes. From a food safety perspective, room and body temperatures favor the growth of many pathogens. That's why proper refrigeration or heating is so important in preventing foodborne illnesses. Effect of pH. Most bacteria prefer a near-neutral pH around 6.5 to 7.5. This is similar to the conditions found inside the human body, which is why many of our resident and pathogenic bacteria thrive there. Acidophiles thrive below pH 5 and alkaliphiles above pH 9. They are specially adapted to these extremes. For example, some bacteria live in highly acidic hot springs or very alkaline lakes. pH affects enzyme activity, transport, and the proton motive force. Enzymes have optimal pH ranges, and the proton gradient across the membrane, which drives energy production, is highly sensitive to acidity or alkalinity. Buffers such as phosphate or tris are often added to growth media to stabilize pH during bacterial growth. In preservation, lowering or raising pH inhibits many microbes. This is the principle behind pickling with vinegar or using lime in certain foods. Oxygen requirement. Obligate aerobes require oxygen for respiration. They use it as a final electron acceptor to generate energy efficiently. Obligate anaerobes are inhibited or even killed by oxygen because they lack the enzymes like catalase and superoxide dismutase needed to detoxify reactive oxygen species. Facultative anaerobes can grow with or without oxygen, but they grow best when oxygen is present because aerobic respiration yields more energy. Microaerophiles prefer low oxygen and often elevated carbon dioxide levels. Environments like the stomach lining or certain soil layers provide these conditions. Aerotolerant anaerobes can tolerate oxygen but don't use it for respiration. They rely solely on fermentation for energy. Understanding these categories helps us culture bacteria correctly in the lab and understand where they live in nature. Water activity and osmotic pressure. Water activity, abbreviated AW, measures the amount of available water in an environment. Most bacteria require a water activity of at least 0.91 to grow. When AW is low, due to high salt or sugar content or drying, it causes osmotic stress and plasmolysis, where water leaves the cell, leading to growth inhibition. That's why salting, sugaring, or drying foods are effective preservation methods. They essentially suck the water out of microbial cells. Halophiles and halotolerant species are adapted to high salt environments like the Dead Sea or salted fish. To cope, cells accumulate compatible solutes such as proline or betaine, which balance the internal osmotic pressure without interfering with cell metabolism. We control water activity through packaging, humidity management, and adjusting salt or sugar content to preserve food and other products. Nutrient availability. Macronutrients, carbon, nitrogen, phosphorus, sulfur, oxygen, and hydrogen are needed in large amounts to support energy production and build biomass. Think of these as the major building blocks of proteins, nucleic acids, and cell walls. Micronutrients and cofactors like iron, magnesium, manganese, zinc, and various vitamins are needed in trace amounts but are essential for enzyme function and other cellular processes. The limiting nutrient, the one in shortest supply, controls the growth rate and final yield of a bacterial population. This is a key principle in ecology and biotechnology. Auxotrophs are mutants that require specific growth factors such as certain amino acids or vitamins that they can no longer synthesize themselves. Finally, the carbon to nitrogen to phosphorus balance and thoughtful media design shape bacterial physiology and even cell size, influencing everything from lab experiments to industrial fermentation. Thank you from Mootechie. We hope this overview has helped clarify the key factors that influence bacterial growth and given you a useful framework for your studies. Thanks for joining and keep exploring the microscopic world around you.